We listed some things about those 12 cranial nerves, and let's see if you can help me remember these things from last time. I said there were going to be 12 pairs of cranial nerves that we have to know. You have to know the names of the cranial nerves as well as what they do, and there were a couple of acronyms that allowed us to remember those. I'm just going to list the 12 cranial nerves. And we had some G-rated versions of these acronyms that allowed you to remember the names of the 12 cranial nerves. We said one of them was O, to touch and feel very green vegetables. Which is weird, but, or O, to touch and feel very good velvet. Which, if you take each of those letters, that would give us O, to touch and feel very green vegetables. Which is weird, but again, we've got the letters there, so now we can go through and list. The first cranial nerve is our olfactory nerve. The second one we said was our optic nerve. The third was oculomotor. Our fourth was the trochlear nerve. The fifth one was the trigeminal nerve. The sixth was the abducens. Number 7 is the facial nerve. Number 8 is our vestibulocochlear nerve. Number 9 is the glossopharyngeal. Number 10 is the vagus nerve. Number 11 we call that the accessory nerve. And the 12th one is hypoglossal. And if you wanted to put the Roman numeral olfactory nerve, that's Roman numeral 1. Optic, you'll see Roman numeral 2. Ocular motor, you'll see Roman numeral 3. Trochlear, you'll see Roman numeral 4. Trigeminal, Roman numeral 5. Abducens, we've got Roman numeral 6. Facial is Roman numeral 7. Vestibulocochlear, we've got Roman numeral 8. Glossopharyngeal is Roman numeral 9. Vagus is Roman numeral 10. Roman numeral 11, and then Roman numeral 12. So we can recognize them this way. We also did the other acronym that allowed us to know which of these cranial nerves were sensory, whether they were motor, or whether they were both. So to add to that, we said there was the other acronym, some say marry money, but my brother says big brains matter most. So we've got that, and this is a good idea of a brain dump that you can do on the back page of the exam. Now, as you go through and you answer these multiple choice questions, you've gotten the names of them down. You know which ones are motor, which ones are sensory, which ones do a little bit of both. And then we even did this diagram that allowed us to know some of the functions of each of these. So we took the facial nerve, number 7, and we just kind of made these little boxy edges of a face. We took the olfactory nerve and we made it the nose because it does olfaction or smelling. We made number 2's the eyeballs because it's the optic nerve for vision. Number 3, we made kind of an eyelid on top of the number 2. It's the ocular motor nerve. It's going to help constrict or dilate the pupil, raise the eyelid. Number 4 we said was the trochlear nerve. It's going to be for moving the eye inferiorly. Number 5 was this big facial nerve. So we let that be a big number 5, kind of representing some of the facial expressions and sensory touch on the face. Number 6 we said was the abducens. That allowed this lateral movement of the eye. We've already got 7 on there as our facial nerve. 8 is the vestibulocochlear nerve, so it represented the ears. So hearing, equilibrium, and balance will be accomplished by this vestibulocochlear nerve. Glossopharyngeal, this picked up taste on the posterior 1/3 of the tongue. We said the anterior 1/3 of the, let me say that again. The anterior 2/3 of the tongue, the taste sensation is picked up by this, once again, this big facial nerve. But the posterior 1/3 of the tongue, sensory information is picked up by this glossopharyngeal. The vagus nerve, we said this was the vagabond nerve, the wandering nerve that wandered into the thoracic and abdominal cavity, into a bunch of other visceral organs like the heart. So we have it wandering down into the heart. Accessory nerve was the one that allowed us to shrug our shoulders. So we'll put little 11s representing the shoulders. And then 12 was the hypoglossal. This is the motor nerve that you would use to control the movement of the tongue. So it's not picking up taste sensation on the front or the back of the tongue. It's actually coordinating muscle movement of the tongue. So to show that, I'm just going to get a different color and write a 12 in there, indicating the movement of the tongue is carried out by this hypoglossal. Okay, let's see if we can leave this up for just a little bit, and I'm going to I'll try and add some detail about the other diagram that we did last time, since we're on this little bit of a review. We tried to sketch the autonomic nervous system last time. This was the autopilot that had the two settings of sympathetic versus parasympathetic. We compared this autopilot to the somatic nervous system, the one that you're in voluntary control of. Let's separate this. In this somatic division, we're just going to see one motor neuron versus this autonomic division. Let's squeeze it down here. This autonomic nervous system, the autopilot, there were always two motor neurons. Let me continue this little line on out. We'll go all the way. How about that? And then I'm going to let this first line, anything on this line will be our central nervous system, so inside the brain and spinal cord. Anything on the other side of this line will be out here in the peripheral nervous system. And then at some point, we're going to run into our different types of effectors. The somatic division, let's compare, there was only one motor neuron in the somatic division. Let me get my colors. I guess we'll use a red color here. Somatic division, again, one motor neuron. This is voluntary. So we said the cell bodies of these multipolar neurons. Again, it's a motor neuron, so we know it's multipolar. It's got its cell body in the brain or spinal cord, and its axon is going to make its way out into the peripheral nervous system. We'll make it all the way over here where it's going to dead end close to that effector. We said the one motor neuron in the somatic division sends rapid signals from the cell body to the axon terminal. And what allows that rapid signal are these heavy layers of myelin. Heavy layers of myelin. Maybe we can just label parts of this neuron. We can see the dendrites, the cell body. This is the long axon. The axon portion is sometimes also referred to as a nerve fiber, right? Because we're going to be talking about these fibers that are going to be releasing at the end different types of neurotransmitter. This is a fiber. If we look at the end of this particular fiber, we see vessels that contain acetylcholine. So that tells us this is what we would call a cholinergic fiber. Cholinergic fiber because it's producing and storing and about to be releasing acetylcholine. Okay, let's add just a little bit more to that. Since we're talking about this somatic division, we know the effectors of the somatic branch are always going to be some type of skeletal muscle. And when we were drawing skeletal muscle last time, we knew that skeletal muscle were these kind of long cylindrical shaped cells, and there's a certain region on that muscle cell called the motor end plate. A little better. This motor end plate, that's the location on this muscle cell where you find receptors for acetylcholine. So this neuromuscular junction was always between what we considered the axon terminal and this motor end plate. We saw at one point where there was a series of events that started with calcium moving into the axon terminal, and that led to release of these neurotransmitters, and they're going to dock to these, again, non-specific ion channels. The effector for the somatic division are always these skeletal muscles. So let me label this as our skeletal muscle. But if we're thinking about the effectors of this autonomic branch, our autopilot, we said it doesn't go to skeletal muscle at all. Instead, I was putting up a box over here that I just labeled collectively as this box represents our cardiac muscle. It represents our smooth muscle. It's going to represent our glands, like sweat glands and digestive glands, and it's going to represent adipose tissue. So these effectors are going to be lined with receptors as well, and I think we listed those. Yeah, I'm trying to think how crowded do we want to get this diagram right off the bat, but you're right. If we're thinking about the receptors that are on here, maybe I can draw them this way, where there are receptors on these targets that are, I'll draw in a blue color because they're receptors for acetylcholine. And that is correct. We call these muscarinic. Muscarinic receptors are receptors that bind acetylcholine and are located on cardiac muscle, smooth muscle, glands, and adipose tissue. We saw that those same targets, they have adrenergic receptors as well. I'll put these different types of adrenergic receptors along the bottom. We saw, it's terrible. I'll draw that. There we go. Alpha 1, we got alpha 2, we saw beta 1 and beta 2 as well as beta 3. So there's a list of different adrenergic receptors, and I think at the end of the last lecture we started to talk about where some of these different types of adrenergic receptors were located. The case study today takes us back to that. So just a reminder, we've got the target organs of the autonomic nervous system. They're dually innervated. So the two different settings, the sympathetic can trigger these adrenergic receptors, and parasympathetic is going to trigger these muscarinic receptors. Real quick, we do see these receptors up here that are also binding acetylcholine, but they're not on these target organs. They're on the motor end plate. Does anybody remember what type of receptor that was? Did I hear it? Nicotinic receptors. So these receptors, I'll just list them over here. These are nicotinic receptors. Maybe I can get that line moved out of the way just a little bit. Our class of cholinergic receptors. We said there were cholinergic receptors and there were these adrenergic receptors. Of the cholinergic receptors, there were two varieties. There were either these, what we called muscarinic cholinergic receptors. Muscarinic tells you their location. Cholinergic tells you that they're binding acetylcholine. So nicotinic is also a cholinergic receptor. We can see it binding acetylcholine. What makes it not a muscarinic receptor is it's not located on these targets. Instead, nicotinic receptors, we said, are located in 3 places. One of them is the motor end plate, and there are 2 other ones that we're about to draw here. Those are the only receptor types we have to know, the variety of different adrenergic receptors, and then the 2 versions of cholinergic receptors, either nicotinic or muscarinic. Let me grab some other colors. The first one that we're going to draw is let's try parasympathetic. So the parasympathetic branch, we're going to see that these neuron cell bodies that I'm about to draw, these multipolar neurons, they're going to have their cell bodies and their dendrites in what we would consider either the cranio or sacral division of the brainstem or spinal cord. Craniosacral. So as we see, if we follow this cell body away from either the cranial or sacral region, it's going to make its way all the way out close to these effectors, and then we're going to see this first neuron come to a dead end. There's always 2 motor neurons when we're trying to draw this autonomic branch. The second motor neuron we're going to see is very short, and it's close to the organs that it innervates. So there we go. We've got our 2 motor neurons. In the autonomic branch, we said it's the first motor neuron that is the only one that has any myelin whatsoever, and it's a very light layer of myelin. And this post-ganglionic neuron has no myelin at all. So that kind of reminds me, now that we've got 2 of these neurons, we've got an opportunity, we've got what we have a collection of these cell bodies outside the brain and spinal cord. Our collection of neuron cell bodies out in the peripheral nervous system we call the ganglia. And we will learn that in the parasympathetic division we call these terminal ganglia because the ganglia are really close to the organs that they innervate. They terminate really close to this particular ganglia, so terminal ganglia. Okay, so we've labeled both neurons. Let's now add to it which neurotransmitters they release. Remember that it's always the case that the presynaptic neuron is going to be storing and releasing acetylcholine. Blue in this case is going to represent my acetylcholine. So once again, we can call, it's already labeled there, we can call this axon portion of a neuron, that's a cholinergic fiber. Fibers, again, nerve fibers are the axon portions of a neuron, and if we follow this axon portion of a neuron, it's producing and storing acetylcholine. So it's a cholinergic fiber, it's releasing acetylcholine, and that acetylcholine is going to land on these receptors, these chemically gated channels, either on the cell body or on the dendrites of this postsynaptic neuron. What type of receptors are those? I mean, it's docking acetylcholine, so we know it has to be a cholinergic receptor. Which one of those 2 cholinergic receptors do we find on postganglionic cell bodies and dendrites? It's another example of our nicotinic receptor. I'll squeeze it in down here. And nicotinic receptors. We'll find them on postganglionic cell bodies. We find them on skeletal muscle motor end plates. And then there's one more place that we're going to find them on. That's going to take us into the sympathetic division. But before we go to sympathetic division, again, the 2 motor neurons in parasympathetic. The first one is releasing acetylcholine. And the second neuron here, if we look at the second nerve fiber, this is also what we would consider a cholinergic fiber. Cholinergic fibers are storing and producing acetylcholine. So even if we go to the think I was able to say that right. Even if we go to this postganglionic neuron and we look at the axon terminal of this postganglionic neuron, it too is producing, storing, and releasing this acetylcholine. So in the parasympathetic division, both preganglionic and postganglionic neurons are both producing and releasing acetylcholine. We know that postganglionic neuron, it's going to be releasing acetylcholine that are docking to muscarinic receptors. Somatic division, it's docking to nicotinic receptors on that motor end plate. Now, for the last one, I'll put it in an orange color. We doing okay so far? I think I've got just enough room to fit the two versions of our sympathetic division that we're going to draw. I think I'm about to go back on my orange color because that seems like it's going to be hard to erase. Let's try instead a purple color. Much better. So sympathetic division, when we start to draw these neurons, and I'm just going to scoot that up a little bit. This sympathetic branch, if we draw this, we're going to see that these two cell bodies that we draw are going to be coming from the thoracic or lumbar region of the spinal cord. So sympathetic division is sometimes referred to as thoracolumbar because these neuron cell bodies, again, originate either in the thoracic or lumbar region. In the sympathetic branch, we're going to see the first neuron is relatively short. And it stops. There is a second neuron that it communicates with. Our postganglionic neuron tends to be much longer. Well, since we have our cell body of a neuron outside the brain and spinal cord, we've got another one of these ganglia. This particular ganglia that we can label, there are these chain ganglia. I'll just put the area, it would continue on above and below this where you would see these cell bodies of postganglionic neurons. We even saw a model in the lab that showed this sympathetic chain ganglia. Most sympathetic neurons will stop at this chain ganglia. Chain ganglia, this is sometimes referred to as paravertebral ganglia because it parallels the vertebral column. Okay, so we got paravertebral there. We've got the two motor neurons. Once again, the first one is the only one that has any myelin whatsoever, and it's a very light layer of myelin. And this postganglionic neuron has no myelin. Let's add the neurotransmitters. It's even the case for the sympathetic division that the first neuron, this preganglionic neuron, is once again packaging, storing, and releasing this acetylcholine. So what would we call this type of fiber right here? The fiber, the axon of this preganglionic neuron. That's right, this is a cholinergic fiber because it's producing acetylcholine. There is this acetylcholine being released into this synaptic cleft. That would make these receptors on the postganglionic cell body and dendrite that's receiving that acetylcholine. We know it's a type of cholinergic receptor, but specifically which type of cholinergic receptor? Yeah, once again, it's our nicotinic receptor. So nicotinic receptors on postganglionic cell bodies. The thing changes whenever we look at this postganglionic neuron in the sympathetic division. For the first time, if I can use my yellow color, we see a neuron that's going to be storing and releasing some other type of neurotransmitter. This is going to be our first example of an adrenergic fiber. So adrenergic fibers are going to be releasing these adrenaline molecules, which we abbreviate as norepinephrine. We can spell it. Norepinephrine is going to be coming from these adrenergic fibers, from postganglionic neurons. We said there was another pathway for the sympathetic division that was not as common, but some of these sympathetic neurons we're going to see the preganglionic neuron does not dead end in the chain ganglia like we saw before. Instead, some of these sympathetic neurons are going to bypass this chain ganglia. I don't have a whole lot of room here, and I'll say make their way into, oftentimes it's digestive organs. The one that we look at here in this example is the adrenal medulla. So I put this kind of pyramid-shaped gland up here that represented the adrenal gland. There's the outer cortex, and then the middle medulla part. And we said some of those cells in the medulla are modified postganglionic neurons. That means they have receptors on them for acetylcholine. Just like all these these postganglionic neurons have receptors for acetylcholine, these are postganglionic cells. They've got receptors for acetylcholine. We can see the first neuron, our preganglionic neuron, like always, is going to be producing and releasing acetylcholine. So we can call this, once again, a cholinergic fiber. And we can see acetylcholine binding to these receptors in the adrenal medulla. That's our last place where we find nicotinic receptors. So 3 places for those nicotinic receptors are skeletal muscle motor end plate at the top, our postganglionic dendrites and cell bodies, and then also cells of the adrenal medulla have these nicotinic receptors. But just like the postganglionic neurons of the sympathetic division, when these cells of the adrenal medulla are hit with acetylcholine, they turn around and start to pump out a mix of adrenaline molecules. In the adrenal medulla, it's going to come out as a mix of epinephrine and norepinephrine. For whatever reason, it's about 80% epinephrine and about 20% norepinephrine. But these are adrenaline molecules, so I'll draw them here as an orange little dot, and they're not going to dock to muscarinic receptors at all. They're looking for these adrenergic receptors. And we talked about last time, these adrenergic receptors are located in different places. Maybe you might remember alpha 1 is located on most blood vessels. Alpha 2 is located on glands that go to the digestive system. So think digestive system glands. Beta 1 goes to the muscular heart wall that does the forceful contractions of the muscular heart wall. Beta 2, we saw that went to some blood vessels, the walls of the blood vessels that go to coronary, the coronary vessels, the one that go to the heart, and then the blood vessels that go to skeletal muscles. And we saw beta 3 is found on adipose tissue. I'm going to take a step back to see if we've left anything off. We can find which fibers are cholinergic, which ones are producing acetylcholine. We can find which fibers are adrenergic, that are producing those adrenaline molecules. We've separated the location of cholinergic versus adrenergic receptors. We've even separated the 2 classes of cholinergic receptors. Any questions about that? Because I'm going to leave it up there. And did everybody get a chance to grab our case studies from today? Did I miss anybody? Let's.
Beta 1 found again?
Beta 1 is going to be found on the muscular heart wall. So when we were looking at the heart, we said there were the beta 1 receptors that go to the myocardium, the beating cardiac muscle, where beta 2 are found on smooth muscles. Smooth muscles are found within the walls of blood vessels. So we've got this model that shows us within the walls of blood vessels, we've got smooth muscle. And if we think of those blood vessels right here that go to the wall of the heart, smooth muscle can dilate and allow more blood flow to the heart. And that's why when the sympathetic signal is sent, it triggers beta 1. The wall starts beating more forcefully and more quickly. So it's going to need more blood supply. So beta 2 is found in the smooth muscle of those coronary vessels, and that's an inhibitory receptor. So this is going to allow that smooth muscle to relax and dilate. So they're doing two different things, but for the same goal of getting you out of this stressful situation. You know, it was always curious to me why it is when you're in fight or flight, how is it that the sympathetic signals can simultaneously cause some blood vessels to dilate and other blood vessels to constrict? And it has to do with the fact that those different blood vessels have different receptors on them. Most blood vessels have alpha 1 receptors and are going to constrict. However, some blood vessels have beta 2, and those are going to be the ones that dilate. So it's interesting, the sympathetic signal can simultaneously close off blood vessels to the digestive system and open up blood vessels to the skeletal muscles or to the heart wall. If you have that lab list handy, I'm going to the top of where it says Lab 14. This is sensory number 1 for the eye models. This is page number 2. And on this page, you've got number 1, where we're actually looking at structures of the eye. And then down at the bottom of page number 2, next to the number 2, it says there are a list of accessory eye structures. We're going to find these on some of the eye models or on the torso models. And it's really the stuff down there, these accessory structures of the eye, number 2 on the list, that we see in this particular slide. So I wanted to point out a few of these things. We will see the term medial canthus and lateral canthus as referring to either the medial angle of the eye, this is the medial canthus, or the lateral canthus. It's also called the lateral angle of the eye. There is, if I can go to this side view, we can see there is a muscle that raises the eyelid. That muscle that raises the eyelid is the levator palpebrae superioris. It's controlled by the ocular motor nerve. So to raise your eyelids, one of the things that needs to be functioning properly is the ocular motor nerve. It controls that levator palpebrae superioris. Again, muscle that raises the eyelid. What we can also see from this angle, you can see how the surface of the eyeball has this little kind of a mucous membrane that covers the surface of the eyeball. This same mucous membrane also covers the back side of the eyelids. Can everybody see that little film? This is called the conjunctiva. There is, it looks like on your lab list, you just have to be aware of the conjunctiva. There is a bulbar conjunctiva, which is on the lower eyelid, and a palpebrae conjunctiva, which is on the upper eyelid. This palpebrae you can think of as it's a pocket that it prevents any debris from getting around to the back of the eye. I don't have contact lenses, but I remember my cousin who started to put contact lenses on as a kid. She would sometimes lose the contact lens. It would go off the surface of the eye, like off the center of the eye, maybe around to the side. And I remember thinking, oh my God, does that like go back into her brain? Like where does that contact lens go? And it can't get around the side of the eye back into the brains because of these little pockets, these conjunctiva. So the surface of the eyeball connects to the back side of the eyelids and forms these little pockets. That's also a pocket that might catch bacteria and get inflamed, and this is one of the causes of conjunctivitis. So conjunctiva, we can see, is the surface of the eyeball as it connects to the back side of the eyelids. We've seen that palpebrae superioris muscle. Where are we at on the list? Lacrimal gland, if I go to this anterior view, we can see above each eye, on the superior and lateral side of the eye, there is a lacrimal gland. This is the gland that produces tears. We can see when tears are produced, They sheet across the eye at a diagonal fashion. They go from the superior lateral side, where the lacrimal gland is located, and they make their way towards the medial angle of the eye. This medial canthus, if we look at it closely, it has this lacrimal apparatus. There are these two little openings, and if you look at the medial angle of some people's eye, you can actually see these little openings called lacrimal punctum. Those little openings allow for these tears to drain into this nasal lacrimal duct. So on your list, you have a few parts of that. You have the what's just called the lacrimal sac. This point up here, that swollen area, is the lacrimal sac, and then the nasolacrimal duct is what connects this lacrimal sac to the nasal cavity. So again, tears are coming from this lacrimal gland. They sheet across the eye in a diagonal fashion. They make their way into these lacrimal punctum and then fill up this lacrimal sac, where they drain then down into the nasal cavity. This is why sometimes if your eyes are watery, or if you are crying a lot, you'll have to blow your nose, because some of those tears are draining into the nasal cavity. Okay, so then that takes us to the extrinsic eye muscles, and these are all of the muscles that you're going to find around the surface of the eye. They help control the movement of the eye. And on Thursday, we're going to have a class handout where we have to list which of those muscles moves the eye in a certain direction, and that's all lecture stuff that'll be for Thursday. This time, we just want to be able to name those 6 extrinsic eye muscles. And if we look at it straight, face on here, it's easy to name 4 of those muscles. And we have an eye model. If you take off this, which would be the eyelids, you can see the conjunctiva behind it in pink. It kind of just shows us a picture of an exposed eyeball. And we can point to 4 of these extrinsic eye muscles really easily. These are the ones that are going to be called the rectus muscles. There's one on the medial side, so we've got our medial rectus, one on the lateral side, the lateral rectus, one below, which is the inferior rectus, and one above, which is the superior rectus. It's easy to find those 4 rectus muscles. There are 2 that are maybe a little harder to find. These are the oblique muscles. There's an inferior oblique and a superior oblique. It's easy to remember the superior oblique because it's on the superior edge of the eye. It's up there, up top. So maybe somewhere on your lab list, write down that this inferior oblique, you will find the inferior oblique between the lateral rectus and this inferior rectus. So on this eye model, when I'm rotating this thing around, right there, we can see there is a muscle, and it's between this lateral rectus and inferior rectus. This inferior oblique is a clue that tells you which side of this eyeball is lateral. Wherever that inferior oblique is, that's the lateral side of the eye. And then at least on this model, you can only see a little bit of this superior oblique. It's really that little piece of connective tissue that attaches that superior oblique to the top of this eye. If we grab a couple of these other eye models, like this one has a little bit more of the skull to it, you can see that lacrimal sac and that nasolacrimal duct. We can see the lacrimal gland, and you can also see some of those extrinsic eye muscles. I point out this one because you can see a little bit more of that superior oblique. So 6 extrinsic eye muscles. We can find them on the eye models. If we've gotten through all of those, then we are now moving up to the very top of page number 2. Underneath number 1, it says, study these eye models and locate each of these structures. You can see there is a fibrous tunic with a few structures under there that we need to be aware of. There is a vascular tunic with, once again, a list of structures that we need to be aware of. And then a sensory tunic with a few things that we're going to draw. So let's make our way through the rest of the eye stuff. If we take one of these eye models and start to open it up, try to bring out 2 different ones. And sometimes they're missing little things that hold them together. So kind of be careful when you grab onto one of these things. But I'm going to hold up one of these eyes, and we're going to go from the most superficial things that we can see on the surface of this eye, and then we're going to start to peel back the layers. So here on the outside part of the eye, One of the things on your lab list that we have to know is called the sclera of the eye. This white outer layer of the eye, let me get my way over here to the pointer. This white of the eye here is the sclera, the outer white surface of this eye. You can see how the white of this eye starts to wrap all the way around to the anterior part of the eye, and then it becomes clear. This clear anterior portion of the eye is what we call the cornea. So the sclera and the cornea together make up this outer fibrous layer of the eye. Going back here, sclera is the white part of the eye. The cornea is that clear anterior part of the eye where you would try and situate the contact lens. So visually looking at this, we've got our sclera, and then we've got that cornea. But at the top, if we were to somehow peel away that top layer of sclera, what we would see underneath it is this darker layer with those blood vessels. Can you see those little blood vessels that are kind of indented or drawn into this darker red layer? This darker red layer is part of what we would consider the vascular tunic, and that's why you can see the blood vessels there. This is a vascularized layer of tissue. If I can jump back to this slide, this vascular layer, this red layer that we're about to look at, it's going to include what we call the choroid layer, as well as the iris, the colored portion of the eye, and the ciliary body. When we look at the ciliary body, there are two things that grow off of the ciliary body. This is a big structure. It basically has two things that are noteworthy. There is the ciliary process. It's as its name suggests, it's going to be sticking off the edge of the ciliary body. And these processes that stick off the edge of the ciliary body, they hold these tiny ligaments that we will call suspensory ligaments. If I can jump to this picture, those are the suspensory ligaments, and they hold the lens in place. So now we can see this red layer that we were just calling the vascular layer. There is this, what we call the choroid layer. This is where all the blood vessels are. And if we follow this choroid layer around to the anterior part of the eye, look right there where this red layer starts to swell. You can see this kind of swollen area of the vascular layer is what we call the ciliary body. I'm going to go back to this slide because we said the ciliary body has two things that are noteworthy. There is the ciliary process that holds those suspensory ligaments. That right there, those little finger-like projections, those are the ciliary processes that you can see suspensory ligaments dangling from. So there's that. And then if we could somehow look within that ciliary body, there would be smooth muscles that are going to allow the pupil here to either constrict or dilate. So I'm going to go back to here to talk about the ciliary muscle that's found within the middle of that ciliary body, and it's going to allow constriction or dilation of the pupil. So anyway, on this, save that. On this model that I'm holding up, if I can peel back the sclera, what we can see underneath is this darker layer of the eye that's got all of these blood vessels around it. So this is our choroid layer, this darker layer. And around the front side, we can see this one happens to be blue. This is the iris. And then the actual hole itself is the pupil. I know it's not labeled up here. But I believe pupil made the lab list. Let me set that down. Pick up this list. We've seen the choroid layer, this vascularized layer. We can see how that choroid layer becomes the ciliary body that has ciliary muscles and ciliary process attached to it. We've seen suspensory ligaments. The iris, in this picture, looks like the iris is brown. And then the pupil would be that hole that's created that allows light to hit the lens. So let's jump into a deeper layer. If I were to take this model and make our way through this choroid layer and open it up even further, well, the first thing that we see is this big pocket of fluid. We're going to call this vitreous humor. It's like a jelly ball. I'll set it down for a second. And we can see inside this eye, you can see how there's a light tan color. On the slide up here, it's trying to use a yellow color, but this yellow color is what we're going to call the sensory tunic. Going back to this slide, there is a sensory tunic that's the innermost layer of the eye. Two things noteworthy about the sensory layer. There is going to be a pigmented layer of cells that act as a shiny mirror. They reflect light. And then on top of this shiny mirror, this pigmented layer, are going to be several bipolar neurons that are all going to be stacked on top of one another. Together, the stacking of these layers of bipolar cells are going to be what we call the retina. We're going to draw all 5 of those cells in the retina and how they communicate to one another. But let's just look at this sensory layer kind of from a zoomed out perspective. Everything in yellow here is our sensory layer. We can consider this the retina. And notice how the retina, it comes from the optic nerve. So we can see it spread into the posterior part of the eye, and then it kind of starts to spread out around towards the anterior part of the eye. But look at how that retina doesn't make its way all the way to the anterior part of the eye. It stops. And where it stops, it forms this kind of jagged little edge of retina. That jagged edge is called the ora serrata. That is something that made the lab list. And if you look at the edge on the inside of these models, the edge of that retina is kind of a serrated, jagged edge. That again is our ora serrata. What else can we say about this retina? There's this part where the optic nerves and the blood vessel enter the back side of the eye. We're going to learn that this is an area there, since there are blood vessels and nerves that enter the back of the eye, there are no photoreceptors on this particular area. So since there's no photoreceptors on this part of the eye, you can't form an image with that part of the eye. So this is often called the blind spot or the optic disc because we're going to see no photoreceptors located in this region. We're going to see kind of just adjacent to this optic disc, there is an area that's just mapped with a dot on this slide. This dot is called the Macula Ludia. In the center of that Macula Ludia is an area called the Fovea Centralis. The Fovea Centralis is an area where you have the highest concentration of cones. We're going to get into the different types of photoreceptors, the rods and the cones, as something that we'll talk about, or maybe we'll talk about it here. Rods and cones are the 2 types of photoreceptors that we're going to see scattered along the retina. Turns out cones are going to be in the highest concentration right there at the macula ludia. Rods, however, are going to be more concentrated out here around this ora serrata. So rods are more highly concentrated around the perimeter of the retina. Cones are concentrated right there in that fovea centralis, around the macula ludia. So rods and cones are going to be on the retina. They're not evenly distributed. I'll just say cones are going to be for sharp vision and for colors. So cones can help you perceive different colors, and they also help you see sharp edges. So I don't know if you noticed, but there's like a visual acuity chart over here with different letters, and you're supposed to stand a certain space away from that and cover one of these eyes, and then way down here at the bottom of the chart, if you can't see sharp edges, then you can't tell whether that's an F or an E. And when you're trying to see sharp edges like that, I'm trying to get that image to hit right there in the fovea centralis because that's where I have the highest concentration of cones, and that's where I'm going to form the sharpest image if I can get it to hit right in the center of the eye. Again, so cones are for sharp edges, visual acuity. Whereas rods, on the other hand, rods are going to be located around the perimeter of the retina, and they are not for seeing sharp edges. Rods are more sensitive to light or dark. Let me say that one more time. Rods are more sensitive to faint light, so they help you see in the dark. I gave this analogy before. Tell me if this lands with anybody in here. I remember, even now it still happens, but I remember as a little kid having to go to sleep in these rooms where, I remember, trying to go to sleep in these rooms and it would be like the whole room would be dark and I would see like a little faint light out of the corner of my eye. And as I would turn to look at that light, I wouldn't see it anymore. And then as I look away, I could see the faint light in my peripheral vision. I could see it out of the corner of my eye, but again, looking right at it, I no longer can see this faint light. What's going on there is when you're looking directly at something, that image is hitting right here near the fovea centralis. You don't have as many rods there. You're not as sensitive to faint light if you're looking directly at something. But when you look away from something, the images in your peripheral vision are landing somewhere along the edges of the retina. There's less cones out here, but there's a higher concentration of rods. So if you're looking, not directly at something and that image is landing around the edges of the retina, if you have more rods there, you're going to be more sensitive to that faint light. So I wasn't going crazy. It's just proved that the distribution of rods and cones are not equally distributed across the surface of the retina. More rods around the perimeter, and so you're more sensitive to faint light there. What else do we have? If we look more closely at a cross-section through this eye, we can see there's the lens that's being supported by the suspensory ligaments. You can see that ciliary body that I was pointing to. It is broken into two sections. There is the ciliary muscle that does the constriction or dilation of the pupil, and then there's that ciliary process where those suspensory ligaments grow from, and again help hold that lens in place. From this view, we can also see the iris. This is a brown iris, and it is opened enough to allow light to hit this lens. So this opening here would be the pupil. But looking at the lens itself, let's let the lens be the structure that divides what we're going to call the anterior segment from the eye versus this posterior segment of the eye. Anything behind the lens is part of the posterior segment. Anything in front of the lens is part of this anterior segment. You'll notice that the anterior segment has an anterior and a posterior chamber. Just to be clear, the posterior chamber of the anterior segment is still in front of the lens. It's behind the iris, but in front of the lens. So this anterior chamber is in front of the iris and in front of the lens. Both anterior chamber and posterior chamber, anything in front of the lens is part of this anterior segment. Maybe I'm so nitpicky about that is because if we think about where this fluid comes from that fills up this anterior segment, it actually leaks from blood vessels in the ciliary process. The arrows here are indicating that there is leakage from capillaries right there in the ciliary process. And that fluid that leaks out of those capillaries is what produces this aqueous humor, this fluid-filled pocket just in front of the lens. So this aqueous humor is much more fluid or watery-like compared to the fluid that you would find in the posterior segment. If we cut an eye through the posterior segment, really this fluid back here in the posterior segment is like a jelly ball. So it's what's called vitreous humor because it's so viscous, it's gel-like compared to the aqueous, the water-like fluid of this anterior segment. Anyway, about that aqueous humor, it originates in the posterior chamber from that ciliary process. And then that fluid actually makes its way out of the pupil into this anterior segment. So again, into the anterior chamber of the anterior segment, and then drains through this process called the scleral venal sinus. That is a vein that drains away this aqueous humor as fast as it is produced. So this is kind of like that cerebral spinal fluid situation that we talked about. We said that cerebral spinal fluid should be drained away as fast as it's produced. And this is the same case with this aqueous humor. It should drain out of the scleral venous sinus at the same pace it's being produced. If it's not, if however the scleral venal sinus is blocked due to debris or whatever, then you can get a buildup of fluid in this anterior segment. This is what would be caused what I'm describing here is glaucoma. You've got a blockage of the scleral venal sinus. So the fluid that continues to leak out of this ciliary process is going to build up. And if there's an excess amount of fluid in this anterior segment, it's going to put pressure back against that lens, and that's going to push against the retina and could cause pain and damage to the cells in the retina. This is something that we'll see on Thursday, just showing how parasympathetic stimulus causes pupil constriction. Sympathetic stimulus is going to cause pupil dilation. So fight or flight, you're looking like this. Rest and digest, you're looking like that. We know that. So back to this picture. We pointed out the optic nerve where the retina kind of makes its way into the back of the eye. We pointed out this anterior segment from the posterior segment. You can see the posterior segment filled with this large jelly ball, this vitreous humor. We can see the optic nerve coming out of the back of the eye. I'll just hold up this eye model. You can easily see this optic nerve sticking out of the back of the eye model. And if we open it up, you can follow that optic nerve in to reveal wherever that optic nerve enters the back of the eye is where we're going to find the optic disc. And on these models, you just have to look real close. Can you see that little pink dot right there? Verify for everybody else. There is a little tiny pink dot if you look at this retina, and it's not very obvious. They used a pretty similar color pink paint, but that little pink dot is going to be our Macula Ludia. I think we had it labeled right there as our Macula Ludia. It's adjacent to that optic disc. What else? I think that's almost everything from the lab list. We need to look a little bit more closely at the retina. What I like about this picture is it shows that optic disc is not directly in the center of the field of view in the back of the retina. The optic disc kind of enters just off to the side. And that's by, that is advantageous because, like we said, there's no photoreceptors right here where the optic nerve enters the back of the eye. So when you're looking at something, the image is going to be hinting this area in the center of the back of the view that we said has the highest concentration of cones. That's our fovea centralis. I'm about to try and draw, I guess, the different layers of the retina because that's the only thing we have left for eye models on our lab list. What we're looking at here is just the layers of the retina. And the layers of the retina that we need to see there, without listing this stuff on the board, there are a layer of pigmented cells. I said these pigmented cells are going to be like a shiny mirror. So any light that hits these pigmented cells is going to be reflected right back away from these cells. And down here you can see the path that light is taking into the eye. Light would be coming in from this direction, and it's going to bypass what we're going to call ganglion cells. It's going to bypass these bipolar cells. It's even going to bypass the axon terminals of these photoreceptors. That light is going to hit the shiny pigmented layer, and then it's going to be reflected back out of the eye. It's on this direction, when it's going back out of the eye, that it is ever picked up by these photoreceptors. Here's where we're zooming in close enough to see these funny looking cells. These are the photoreceptors. And we can see the cell bodies right there in the middle. We can see on one end of the cell body there is an axon and an axon terminal. And then this kind of expanded end of the photoreceptor is the dendritic end. You'll see how some of these dendritic ends are more or less the same width all the way down, and other dendritic ends are forming this funnel-shaped or a cone-shaped. So just by looking at the dendritic ends, we can tell whether we're looking at a cone or whether we're looking at a rod. So now we can visually tell rod from cone, and seeing how there are way more rods than there are cones, would you think that we're close to the fovea centralis or around the perimeter of the retina?
More rods is going to be on the outside.
That's right, we're around the perimeter of the retina because we have a higher concentration of rods than we have cones. All right. And remember last time when we were talking about neurons, we said neurons are like one-way streets. The signals are always picked up by the dendrites and sent towards the cell body and then towards the axon. So remember, it's interesting. These are the only cells that respond to photons of light, and the only part of the cell that can respond to a photon of light is the dendritic end. So it's interesting that light passes all of these neurons and doesn't cause any of them to fire until it bounces off of this pigmented layer and comes into contact with the dendritic ends of these photoreceptors. Right, so if we can trigger the dendritic ends of these photoreceptors, now we have an action potential that goes through the cell body and down the axon all the way to the axon terminal, and if it's sufficient enough, may trigger these what we're going to call bipolar cells in the middle to send a signal to these ganglion cells. Since you may see this in any direction flipped around, let's try and draw these same things in a direction in which the pigmented layer I'm going to put up top. So this will represent my pigmented layer. Pigmented layer of the retina. We know that these are the cells that act as a shiny little mirror. So, you know, light, this represents, if it represents light, this light is going to come up and bounce off of that pigmented layer. It does nothing at all on its way into this pigmented layer, but once it is reflected back off of that pigmented layer, now is when it's going to trigger some of these photoreceptors. So let's add these photoreceptors into our diagram. We're going to see that embedded into the pigmented layers are going to be the dendritic ends of the rods and the cones. And I'll just use this color, this red color. I'll put a cell body in the middle, an axon and an axon terminal down the other side. Let's squeeze another one next to it. So with the dendritic ends being more or less the same width, I'm trying to draw these rods. Right next to it, I'm going to put one of these dendritic ends that we can see expands into kind of a cone shape. So these are all classified as photoreceptors. And two types of photoreceptors. We can see there are these rods and there are these cones. Both of them are picking up photons. We said cones are sensitive to colors and sharp visual acuity. Rods are sensitive to faint light. They're going to be used in the dark. Rods and cones, all of these here, we can see rods and cones are always going to synapse with these red cells. So just happen to grab a blue color here. The second neurons in this chain of neurons, we're going to see that photoreceptors are always going to communicate to these cells that have a dendritic end. They have their cell body in the middle, and they have a short little axon portion. Structurally, we would refer to these as a bipolar cell. And that's the name that they're given as well. They aren't even given a more descriptive name than that. The cells in the middle of the retina here, we just refer to these as bipolar cells. Bipolar cells are always going to communicate to what we call ganglion cells. And ganglion cells are also a bipolar cell. So there's a dendritic end, there is a cell body, and then there's an axon portion. The axon portion of these ganglion cells are going to turn and kind of do a 90-degree angle turn, and then they're going to make their way out of the back of the eye. I've just drawn one of these ganglion cells, but multiple axon portions of ganglion cells. You can take a bundle of the axon portion of ganglion cells, and this makes up the optic nerve. So the optic nerve is just a bundle of the axon portion of these ganglion cells. Let's label this as our ganglion cell. Sorry if the orange is not coming through. I didn't anticipate that. The orange one is our ganglion cell. And you can see all the axon portions of these ganglion cells kind of making up the optic nerve. Okay, so these three are always going to be communicating to one another. Photoreceptors convert these photons into action potentials and send an action potential to the bipolar cell. Bipolar cells talk to ganglion cells. Let me grab two other colors just to put some other noteworthy things in here. I'll try and use these two different colors. Between the bipolar cells and these photoreceptors, you can see they're using kind of a green color to point to these horizontal cells. So between bipolar cells and photoreceptors, I'm not a great artist, but I'm just going to draw these little arms that show this cell here that we're going to call a horizontal cell. These horizontal cells, they interfere with the sensitivity of bipolar cells. So they can either increase or decrease how sensitive these bipolar cells are to signals sent from the photoreceptors. It's kind of like adjusting the brightness or the contrast on like on a picture or something like that. You can adjust the color slightly. So the horizontal cells do this color and light sensitivity adjustments between photoreceptors and bipolar cells. Then between bipolar cells and ganglion cells, there's another cell that is shaped the same way. It's doing the exact same thing. It's adjusting the sensitivity between bipolar cells and ganglion cells. So functionally, it's doing the same thing as a horizontal cell, but since it's located between ganglion cells and bipolar cells, we're going to give it a different name. These are going to be called the amacrine cells. So together, ganglion cells, amacrine cells, bipolar cells, horizontal cells, and photoreceptors, those are the 5 cells that you see that make up the retina. Let me go back to our slides. We were just right here, and you can see the surface of the retina now that we've kind of stepped back a little bit. All of those multiple layers of yellow structures, those are bands upon bands of the axon portions of ganglion cells. And you can see how they turn and they just go out of the back of the eye. They form this optic nerve. And if we can look closely, we can see if we can make it through the bands of ganglion axons, we get into the cells that make up the retina. All of those thin cells, again, zooming in on that. Ganglion cells, bipolar cells, and then all of these photoreceptors embedded into that pigmented layer. Here's the same picture, just zoomed in slightly. We can again see rods, and then we can see cones. This is just showing that cones are going to come in a variety that are sensitive to different wavelengths of light. Not really important for the anatomy part of this class, but as humans, we have 3 different types of cones. If you're colorblind, you don't have all 3. You might have just 2 types of cones in the body. What's kind of interesting is that there are some birds that have 7 different types of cones in their eyes. So they can see wavelengths that our eyes can't perceive. Kind of crazy. Anyway, so as mammal eyes go, they're relatively unspecialized compared to birds. But
Mark, is that something specific?
what I was attempting to do with these arrows is I was showing kind of like what we're looking at right here, that light actually goes through the pupil, it enters the posterior segment of the eye, and it actually bypasses those ganglion cells, and it bypasses those bipolar cells, and it never triggers them. In fact, it bypasses the axon portion and the cell body of these photoreceptors. They don't respond to light. It's not until that light hits the retina, it's not until that light hits this pigmented layer of the retina that acts as a mirror that reflects that light back the other way. Now that the light is coming back out of the eye, this is the direction when it's actually picked up by the brain. Because the dendritic ends of the photoreceptors are the only parts of these neurons that are sensitive to photons of light. So it's not until those photons of light hit this mirror and are reflected back to the dendrites that they're picked up. It's that's interesting that before these photoreceptors pick up the light, it actually has to go through these ganglion cells, which is why images, the light that has had to go through these ganglion cells and bipolar cells gets refracted and sent different directions. So you wouldn't see as sharp of an image if the light had to pass through those ganglion cells and bipolar cells. Look at this layer of nuclei. These are all the nuclei of the ganglion cells. They more or less line up. Now, if we go another layer deep, look at how all the nuclei of these bipolar cells line up. That's our second layer of nuclei. And now look at our third layer of nuclei, how all of those nuclei of the photoreceptors line up. Keep that in mind, and let's go back to this slide that I jumped over that looks like this. These dots, these are all the nuclei of the photoreceptors, that first line. And then if you look at the dots here in this middle series of dots, these are all the cell bodies of bipolar cells. And then the most superficial layer of dots, these would be the cell bodies of ganglion cells. And again, if the images have the light has to go through the layer of ganglion cells and bipolar cells before it hits photoreceptors, this is going to form more of a blurry image versus an image that doesn't have to pass through as many ganglion cells or bipolar cells. One of the reasons you can form such a sharp image in this macula ludia, as you can see here, ganglion cells and bipolar cells get out of the way. So you have just a high concentration of cones and photoreceptors. So there's less diffraction of the light that has to pass through neurons before it's processed by those photoreceptors. So we're catching back up to here, these photoreceptors. And I just took time pointing this out because there is a model in lab that looks like this. And we can see on this side of the model closest to the door, you're kind of zoomed out. And you can see these large blood vessels, which are part of that vascular choroid layer. The cells at the very base of this model, this is part of the sclera, the white part of the eye. So we can see sclera, we can see the vascular choroid layer, and on top of that sits the retina. If you look closely, you can see pigmented layers of cells, these photoreceptors that we'll call rods and cones. We can see bipolar cells and even those yellow ganglion cells. If I go over to this half of the model, we've zoomed in on just the photoreceptors, so we can see the pigmented layer on the bottom and then either rod or cone embedded into that pigmented layer. So on your lab list, wherever I set mine, there is study the histology model of the retina. We should be able to find, and that's this model, those ganglion cell layers, which are in yellow, bipolar cell layers, which I'm going to call a reddish layer, rods and cones, which are kind of a bluish color, and the pigmented layer, which is a tan color. You can see the nuclei with cells with purple nuclei in them. So from my lab list, that takes me all the way through the structures of the eye. The anatomical term for the earlobe is going to be the auricle or the pinna. Either one is, we have pinna on the lab list, but you could say auricle or pinna. We'll learn what this does, and I probably shouldn't be getting into all of this yet, but what the pinna does is it catches sound waves from different directions and focuses them into the ear. So it's, for example, how you can tell if somebody's calling your name and standing in front of you, that's going to bounce around differently in the ear than if somebody's standing behind you and calling your name. The sound waves bounce around differently in the pinna, and that allows you to tell something about the direction from which those sound waves came from. So catches sound waves, it funnels them into this external acoustic meatus, the next thing on the lab list. External auditory canal or external acoustic meatus. This is where you would stick a Q-tip if you were cleansing your ears. But the sound waves make their way into this external acoustic meatus, and then they run into the eardrum, this tympanic membrane. On your lab list, the tympanic membrane is part of the external ear, but really that tympanic membrane is the dividing line. So on this side of the tympanic membrane, we're in the outside, the external ear. But if we go on the other side of the tympanic membrane, we're now in this area that we call the middle ear. So the middle ear is really going to start with this tympanic membrane, and then it also houses the three ear bones that we call the ear ossicles. There is, if we zoom into that, there is the first ear bone or ear ossicle that we're going to call the malleus. It directly touches the tympanic membrane. Then in the middle, there is the bone that we call the incus. I guess the S got cut off there. Malleus, incus, and then the last one here that is kind of a little stirrup-shaped bone, this is the stapes. Maybe put a little star next to the stapes, because we're going to learn something about the stapes, that if I could somehow pick this stapes up and remove it for a second, what we would see underneath the stapes is an oval-shaped window. So maybe somewhere note that the stapes covers up the oval window. We'll talk about that oval window. If we fall through that oval window, we're going to be in a compartment of gelatinous mass. So if the stapes vibrates, it's going to cause all of that gelatinous mass on the other side of the stapes to also start to vibrate. Anyway, these ear ossicles that we find in the middle ear, their job is to catch sound waves in the air and amplify those sound waves as they make their way into the inner ear. In the inner ear is where we have the nerve for actually picking up those sounds. Before we get to the inner ear, the other thing that we can see part of the middle ear is this structure called the auditory tube. It's also called the eustachian tube is another term it goes by. Auditory tube, eustachian tube, or pharyngeotympanic tube. Either of those terms is usable. Feel free to use whichever one you can spell correctly. But this auditory tube or eustachian tube connects the middle ear to the back of the throat. If you've ever been in like riding up and down in the hill country or up in an airplane where you have to go up high in elevation and then come down in elevation, one of the things that you can do to prevent your what they refer to as your ears popping, what this auditory tube does is it tries to make sure that atmospheric pressure is equal on either side of this tympanic membrane. And like I said, if you're flying in an airplane and you go way up in the sky, then pressure starts to decrease on the outside part of the tympanic membrane. And that means pressure is going to be higher inside the eustachian tube. And one way to balance that pressure out is to have like a nice deep yawn where you stretch your jaws out, and that opens up that auditory tube in the back of the throat and allows pressure to equalize on either side of that eardrum. Chewing gum is also something that you can do to kind of help open that auditory tube and allow pressure to equalize on either side of that tympanic membrane. So anyway, middle ear, we can see the ear ossicles. We know one of those ear ossicles covers up the oval window. There is also a round window that doesn't have any cover on it. We can just see a little depression there that I might as well point out as the round window. And then the eustachian tube or auditory tube connects this middle ear to the back of the throat. Now we get into the inner ear. This is really just the back side of the middle ear. So I'll jump over that for a second so that we can now look at this inner ear, this bony structure. We said one side of it looks like a snail shell that we're going to call the cochlea. And then the other bump over here is not coiled like a snail shell. Instead, what's unique about this bump that we call the vestibule, it has these semicircular canals that are anchored just to that side. So here now we're looking at just the inner ear. We can see this little area that we call the vestibule. It has those two gelatinous masses inside that we're going to call the saccule and utricle. It also has these semicircular canals that are all anchoring to this vestibule. None of those semicircular canals are touching this cochlea over here. Just so that maybe planting the seed for Thursday, we'll see the cochlea is the organ that is accomplishing hearing, while the vestibule and the semicircular canals are conveying information about body position and posture. So equilibrium and balance with the vestibule and semicircular canals, hearing with the cochlea. You can see the lab list, it actually separates the inner ear into what we call the bony labyrinth and the membranous labyrinth. Let me see if I can find a picture. Here's a good picture of just the bony exterior, what they're calling the bony labyrinth. And I'm just looking at this picture here. The bony labyrinth is the hard candy shell. Inside that hard candy shell, if we look inside, we're going to find these gelatinous membrane-like regions. So this is not labeled. This is the bony labyrinth, the outer cochlea. This is the bony labyrinth that we call the vestibule. And these are the bony labyrinths that we call the semicircular canals. I'm going to go back to this picture because now it has made a distinction between the semicircular canal, kind of that outer bony labyrinth, and then these semicircular ducts, which are the membranous, gelatinous inner layer. See how your lab list wants you to know the bony outer labyrinth, the semicircular canal, the vestibule, and then the cochlea, but also know that inside each one, like inside the vestibule, for example, there are those membranous gelatinous masses like the utricle and the saccule. You can see saccule is closer to the cochlea. This utricle is closer to the semicircular canals. So utricle and saccule are these gelatinous masses within the vestibule. You can see semicircular ducts in blue are those gelatinous masses inside the semicircular canals. And then inside the cochlea would be the outer bony labyrinth. Inside the cochlea, there is this, what is shown in purple here as the cochlear duct. Or they're showing it, I guess, in a blue color is the cochlear duct. We're going to see how it's like a spiraled garden hose. We're going to take a cross section through that, maybe coming up next. In addition to those semicircular ducts, which are kind of in the center of the semicircular canal, look at how those semicircular ducts, they swell up right as we get close to the vestibule. These swollen areas of the semicircular ducts are called ampulla. And within those ampulla, this is where we find specialized receptor regions. In fact, in each of these gelatinous materials, we find a specialized purple receptor region. Here's where we're kind of getting into some of the details of Thursday's lecture. Inside those ampulla, we're going to find a specialized receptor called the crista ampullaris. I'm looking to see. So maybe not on the lab list, but I can tell you for the lecture list, crista ampullaris is going to need to be something we remember. So I'll point at it again on Thursday. But what the crista ampullaris does is it conveys information about rotational direction. So if you are like the gymnasts that jump in the air and do all those back flips and then land on their feet, how they know when to come out of that tuck and land on their feet. This balance, what's called dynamic equilibrium, how they can tell where their feet are even when they're spinning in the air. This is accomplished by those receptors called the crista ampullaris. So like one of those ice skaters that does the twists in the air and then knows how to land, that dynamic equilibrium all comes from the crista ampullaris. When we look at this on Thursday, we'll see that each of those, I'll go back to this slide one more time, each of these receptor regions, whether it's the crista ampullaris or these receptor regions in the vestibule, within the utricle and within the saccule, there are these purple regions which are receptors as well. These receptors are called macula. They're going to be conveying information about what we call static equilibrium. So dynamic equilibrium is if you're flipping through the air and you want to know when to come out of that tuck to land on your feet. Static equilibrium, which is monitored by these macula, this is how you're able to tell, like if you're in the elevator with your eyes closed, you can tell by way of these macula whether the elevator is going up or going down. Or if you're in the car with somebody, you can tell whether you're accelerating or whether they're hitting the brakes due to the macula. Anywhere where we see these receptors, there are going to be hair cells kind of embedded into this gelatinous material. And as we will learn on Thursday, hair cells, if you bend the cilia in one direction, they get excited. And if you bend the cilia in the opposite direction, it prevents them from sending a signal. I thought I had a slide in there about hair cells. There it is. I skipped over it. We said hair cells we will look at them more closely on Thursday. Just know that if you bend the hairs in one direction, it allows an influx of positively charged ions. And if you bend them in the opposite direction, it closes these gates and it prevents any ions from moving in. Not to cause you to pull all of your hair out, but I'll say something. Where were we? Here we are. There's something interesting about the endolymph when we draw the hair cells later. We're going to see all of these hair cells are going to be embedded into this fluid that we call endolymph. This picture shows us that, and without causing a bunch of confusion, you would have to imagine that each of these hairs are embedded into this gelatinous endolymph. Endolymph has an extremely high concentration of potassium ions. There's more potassium ions outside of hair cells than there are inside of hair cells. Remember, a depolarization is just when a cell becomes more positively charged. In our previous discussions of depolarizations in neurons, sodium ions rushed in to depolarize that cell, to cause the cytoplasm to become more positively charged. Again, we won't see any questions on this, but I just wanted to point it out in case you caught it here. In hair cells, they're embedded in endolymph, and this is one of the unique places where it's actually an influx of potassium ions that cause depolarization. Remember, the cell doesn't care whether it's sodium or potassium. The cell just cares, am I taking on positive charges? If I am, then I'm depolarizing. Am I getting rid of positive charges? If I'm getting rid of positive charges, then I'm repolarizing. We can get into how the sodium-potassium pumps work to make an excess concentration of potassium on the outside. But we know if there's a bunch of potassium on the outside and very little potassium inside, then as soon as the gates open, potassium is moving in. If the cell depolarizes, again, if it depolarizes past threshold, just like we saw in all axon terminals, voltage-gated calcium ion channels open, calcium ions move in, and that's what leads to the release of neurotransmitter. If enough neurotransmitter is released, we're going to get an action potential down that postsynaptic neuron. Anyway, hair cells, they're embedded in endolymph, and if they bend one way, they depolarize, and if they bend the opposite way, they're prevented from depolarizing. We find these hair cells in each of these receptor regions. So on Thursday, we're going to look closely at the hair cells in the ampulla, which are for, like we said, dynamic equilibrium. We'll look at hair cells in the macula, which are there for static equilibrium. If you're in an elevator and you want to tell whether you're going up or down, or if somebody's slamming on the gas or slamming on the brakes. So everything is about equilibrium and balance over here in the vestibule and semicircular canals. But if we get over here to the cochlea, this, what we call the organ of Corti, this purple spiraled staircase looking thing, this organ of Corti is the organ that perceives sounds. So when we zoom into this organ of Corti, just to try and show what those slides are in the back of the room, we've taken this spiral-shaped cochlea and we've just taken a cross-section through it. And like I said, that cochlea is like a coiled garden hose. And when we take a cross-section through that garden hose, we can see all of these different sections of that hollow tube. And unlike a normal garden hose, we can see that this garden hose is basically separated into 3 different compartments. If we zoom into just one of these coils of the garden hose, so here's the hollow center of this specialized garden hose. Like we said, it's subdivided into 3 different segments. This middle portion is going to be what we call the cochlear duct, and then it is flanked on either side by either the vestibular duct or the tympanic duct. Some textbooks will just call this the vestibular duct, which I think is what your lab list does. You're welcome to call it vestibular duct. Some lists will call this the scala vestibuli. Either one is usable, so use whichever one you want. So scala tympani or tympanic duct refers to this hollow chamber down here. I'm sure we're going to draw this on Thursday, so maybe save that for Thursday. In your class handout that you'll get Thursday, this is already drawn for you, and you just have to label it. But what we will see, let me grab that inner ear model. On the lab, you should be able to find both of these larger ducts, our vestibular duct and our tympanic duct. The duct in the middle is the cochlear duct. It's easy to find. But here's how you can tell vestibular duct from tympanic duct. You can see this thin little membrane here. That thin little membrane is going to be what we call the vestibular membrane. We can see it right here, this thin little vestibular membrane. It's always between the vestibular duct and the cochlear duct. So easy to find this thin little vestibular membrane. Now we've labeled this vestibular duct. We know cochlear duct's in the middle. So down here is our tympanic duct. In addition to this vestibular duct, you'll notice there's another little membrane that is sticking off kind of in the middle of the cochlear duct. I'm going to zoom in on this membrane. It's going to be called the tectorial membrane. Here we've zoomed in on this tectorial membrane. It's this little hinge of connective tissue that's basically just delicately sitting on top of these hair cells. If you look closely on this model, you can even see the hair, the little bristles of these hair cells. And looking closely, you'll see 3 of these outer hair cells and 1 inner hair cell. All of them have the little cilia that stick up right close to this tectorial membrane. If this let me hold it this way. If this vestibular membrane starts to move a little bit, then that's going to cause the waves to start to jostle around in this cochlear duct. And if this vestibular membrane moves sufficiently enough, it's going to get this tectorial membrane to start to wobble back and forth. And this tectorial membrane just bounces up and down slightly. It's going to trigger these hair cells. And those hair cells, if triggered, are going to send a signal back to the brain that is perceived as sound. Notice that the hair cells are sitting on top of this basilar membrane. So on top of the basilar membrane are hair cells. On top of the hair cells is the tectorial membrane. On top of the tectorial membrane, if we went up a little further, that's where we would find that vestibular membrane. We can see them all here. This thin vestibular membrane, there's the tectorial membrane on top of the hair cells, and then under the hair cells, it has a number 6 on this model. There's the basilar membrane. So we'll see that basilar membrane. Underneath that basilar membrane takes us down into the tympanic duct. Not that we really have to know the difference between inner hair cells and outer hair cells, but it's really the inner hair cell that's the sensory neuron that perceives most of the sound. Those outer hair cells, they basically can enhance that response. And here's what's going on in that analogy. If you've ever been in a loud concert or something like that, and you're trying to have a conversation with somebody right next to you in a really crowded, loud room, what you're having to do, if Evelyn's trying to say something to me and we're in a really loud room, like we're at a concert, I can hear the music from the concert, but I have to tune out the music from the concert to really kind of focus in on what she's telling me. So if you've ever been in one of those situations where you're trying to tune out some noises and focus in on particular noises, it's these outer hair cells that can kind of fine-tune which noises you're trying to focus on. The inner, again, this is more detail than we'll have to know. Inner hair cell perceives the sensory information. Outer hair cells can adjust the sensitivity to certain sounds that you're trying to pick out. We just have to know all hair cells are responding to movements of the tectorial membrane, and if triggered, they're going to be picked up, sending signals back to the brain perceived as sound. From this cross-section of the ear model, look at how we have this part that goes over to the vestibule. This is the vestibular nerve, and then this would be the cochlear nerve. And if you go back to where they have not yet branched, this would just be collectively called the vestibulocochlear nerve. On our large ear models, both of them you can see these sensory nerves where one goes over to the vestibule. This would be the vestibular branch. And then the other part of the nerve goes just to the cochlea. So that would be our cochlear branch. So we can see vestibulocochlear nerve. And you'll notice that the vestibular branch has this ganglia on it. Only present on the vestibular branch and not the cochlear branch. Then there is a note at the bottom of page 3 that talks about the ductus endolymphaticus, which is not something I'm going to point to on the lab list, but that is referring to this gray sac of gelatinous mass that you see kind of hanging off of these inner ear models. So there's a note about it, but I won't point to that. Vestibulocochlear nerve, we just pointed out either the vestibular branch that has this ganglion or the cochlear branch that does not have that ganglion. Then that cross-section of the model that looks just like this, like I said, we have to recognize the vestibular duct, the vestibular membrane, the cochlear duct, this tectorial membrane, the tympanic duct, and then right there, this basilar membrane that separates the cochlear duct and the tympanic duct. If we zoom into those hair cells, you can see the hair cells, the 3 outer hair cells and the 1 inner hair cell. Between the hair cells, you can see what are called supporting cells. They don't have hair, they don't perceive any kind of sound. They just hold up and make sure those hair cells are where they're supposed to be. They support those hair cells. For whatever reason, maybe make this note on the lab list. You can see on the lab list underneath cochlear duct, there is the term where it says organ of corti. This whole picture that we're looking at right here is the organ of corti. That is not something that I can just stick a pin in, because the organ of corti is composed of those hair cells, both the inner and outer hair cell. It's composed of this tectorial membrane as well as that basilar membrane. So as long as we know those parts, as long as we know tectorial membrane, hair cells, maybe those supporting cells which are around the hair cells, then this basilar membrane that separates the hair cells from this tympanic duct. We've seen basilar membrane, tectorial membrane, scala tympani or tympanic duct. Maybe add to that, if I go back to this, know that the vestibular duct or the tympanic duct are going to be filled with a fluid that we call paralymph. And it's only the cochlear duct in the middle that's filled with a different type of fluid called endolymph. We'll make some notes about endolymph on Thursday. One of those crazy noteworthy things is that endolymph has an extremely high concentration of potassium ions. Not that that's going to be big in the story, but we'll see that potassium rushes into those hair cells to cause them to depolarize. That's unique to the other depolarizations we talked about. That's it for the lab list. So I'm just going to answer some questions now and go through the anatomy stuff. No problem. Yeah.